Jinglei Lv. So currently I'm a senior lecturer in the School of Biomedical Engineering at the University of Sydney. So my research is about neuroimaging, particularly functional MRI, diffusion MRI, and multimodal fusion. So I develop a neuroimaging methods for neuroscience research as well as research into brain disorders. And so today I'm going to share with you our experience about building up a tissue-unbiased multimodal brain and template. And it also comes with a tractogram template and the application of this template. So firstly, let's start with brain template. So if you work in the field of neuroimaging, you must use the brain template a lot. So a brain template is a reference of the healthy human brains, or sometimes a brain template can be built by the disease group as well. But usually it's a reference of the healthy brain. It reflects the commonality of the healthy brain, and it provides correspondence across subjects. And it is also a standard space for statistical analysis. So if we register all the subjects to the template space, we can compare individuals. We can also compare groups. This helps us find imaging biomarkers for a certain disease. So on the right top, you're looking at the MNI 150-5 anatomical template. So as we know, it is built solely based on the T1-weighted image. I think in certain versions, there are also T2-weighted images. But anyway, so it is about it's a template about brain anatomy. So you could see that it provides quite nice details about the folding patterns in the gray matter and the subcortical nucleus. But if you look at the white matter, there's quite limited information there. Well, I think this is because of the limitation of the T1-weighted imaging modality. So if we look at the bottom right, so this is a template called fiber orientation distribution template. So this template is built based on the diffusion-weighted MRI. And the method itself was firstly proposed by Raphael in 2016. So initially, this template is designed for the pixel-based analysis. So if we look at this template, so it provides quite nice information about the white matter microstructures. So for each location, we can calculate the fiber density. We can estimate the fiber orientations, and for certain regions, we can also find rich information about the crossing fibers. And however, if we look at if we look at the superficial white matter, or the regions which is close to the gray matter, or even in the gray matter, I think it's easily to see that we lost the specificity. We lost the folding pattern, and we got quite blurry about the gray matter white matter boundary. So that's why we feel that each template is kind of biased to different tissue information. So then we ask, why not we combine different modalities? We use the complementary information from each modality. Then we build up a coherent template that is not biased to any tissue. So next, we propose to use multimodal registration to build a tissue-unbiased template. So in our initial idea, we propose to use to combine the T1-weighted, T2-weighted image and also the diffusion matrix like the MD, FA, and on the right, we have the apparent fiber density and fiber complexity calculated with a matrix. So we propose to combine them, and we developed an iterative pipeline to optimize the template. So the method itself is based on symmetric group-wise normalization method, and a few commands were from the ANT toolbox. So we built up this pipeline, and we adopted the data set from the Human Connectome project because it provides quite high quality, large sample, multimodal data. Well, it includes a very high resolution structure MRI and very high multi-shell, multi-angular resolution diffusion MRI, and also high quality FMI. So this is the result we get. So this multimodal brain template has the anatomical component on the left and the diffusion components on the right. And most importantly, so all the components are in the same space, which means that if you look at one single work cell, there's correspondence across all these images. And so we can also because through this template generation, we also got very nice optimized multimodal registration, and we apply this registration to the fiber orientation distribution, and we averaged it across all the subjects. So later we generated the FOD template. And based on this FOD template and the anatomically constrained fiber tracking, we also generated the tractogram template. So firstly, let's look at the anatomical components. So we compared our multimodal template with the MNI template. So just qualitatively, we found that our template provides quite a nice tissue specificity, which is equivalent to the MRI template. And yeah, so based on the T1 weighted component, we can do surface reconstruction. So here you're looking at the white matter surface reconstructed from the multimodal template. And we can also generate a very nice tissue segmentation, including grey matter, subcortical region, the white matter, and the CSF. And well, this segmentation will be used to constrain the fiber tracking later when we generated the tractogram template. So next, we're going to look at the FOD template, because we believe FOD template has the most rich information about the diffusion component. So on the left, we have the multimodal template, and on the right, we have the single modal template. As we can see that our multimodal template provides good specificity in both deep white matter and the superficial white matter. So particularly now you can see quite nice folding patterns of the gray matter, and we also see very nice white matter gray matter boundary. So this is a very important improvement comparing with the previous single model template. And we did develop the quantitative measurement to evaluate the template. So here, what we measure is the individual template difference. So one metric is FOD, rooted mean squared error, and the other is the angular correlation. So in this red blocks, we have all the multimodal approaches, and on the right we have the single modal approaches. So clearly we can see that the multimodal approach improves the individual template alignment in the superficial white matter, in both metrics. So we also have some quantitative comparison, and well, through this comparison, we found that this T1, T2 plus MDFA method give us the best performance in terms of the individual template alignment for both the deep white matter, this green regions, and the blue regions, which is the superficial white matter, and also the whole brain. And another evaluation was done through the tract segmentations. So again, we also mirror the individual template alignment, and well, this green distribution, light green distribution shows the individual template alignment for the single modal registration, and these other colors indicate the individual template alignment for all the multimodal methods. Again, we found that the T1 plus T2 plus MDFA method give us the best alignment in terms of the bundle segmentation alignment, and also the green matter ending point alignment. So in conclusion, we did show that the multimodal registration provided a lot of advance in the brain alignment, particularly for the white matter and the green matter alignment. And next, so we use the tissue segmentation generated from the structure components, and we use it to guide the fiber tracking based on the FOD template. And the method we use is anatomically constrained fiber tracking. So with this, we generated the tissue-unbiased tractogram template. So yeah, the advantage of this template is that all the fiber ends are well aligned with the green matter white matter boundary and the subcortical nucleus. This is very important for joint structure and functional analysis, and the joint white matter and green matter analysis. Yeah, we will show some examples later. So yeah, there's a concern that averaging the FOD across subjects may cause some spurious fiber tracking in the tractogram template. So we proposed a filtering strategy to optimize the tractogram template. Here, the idea is that we inverse warp the tractogram template into the individual space, and then we use the individual fiber orientation distribution to filter the template. And in the end, we have a voting strategy to find the most commonly reliable fiber trackings. So we got the refined tractogram template. The filtering method is called the safe spherical deconvolution informed filtering. Okay, so now this is about the paper of building a tissue-unbiased multimodal brain template. So we have open sourced all the code and also the result of this paper. So welcome to use it. Welcome to test it. If you want to build up some study-specific template, welcome to contact me. I'm happy to collaborate. And next, let's look at some applications of the multimodal template. So firstly, so this is the work we presented in the last ASK-MR meeting. Here we combine the diffusion MRI and the functional MRI based on the multimodal template, particularly the tractogram template. Here our aim is to map the functional role of white matter. So if you are familiar with the functional brain mapping, the functional MRI is a major modality. So usually you design a task and then you record the fMRI both signals during the task. Then you compare the brain signals with the task design. Usually with the generated model, you can find the best fitting, and so then you can declare the task actuation as this red or yellow regions. We all know that the white matter connectivity play a very important role in the brain function, but there rarely are tools to map this role of white matter in the brain function. So to address this gap, we propose the Tract-DFC to mirror or to associate the brain functions to the white matter tracts. Here's the idea is that for each white matter tract, we can find the gray matter voxels it connect, and with the gray matter voxel location, we can find the corresponding fMRI time series. Here we simply apply multiply the two signals, then we generate one single time series. We call it the Tract-DFC. So this signal is associated to each white matter tract, and it reflects the functional co-fluctuation or the covariance of the gray matter regions this tract connect. So this definition actually borrows the idea of the edge-centric functional connectivity from Fatzkowitz in 2020. So on the right, I'm going to show an example of the Tract-DFC calculated with the HCP resting state fMRI. You can see that the rich dynamic patterns there. We can calculate the Tract-DFC for resting state as well. We can also calculate the Tract-DFC during the task. Then we can compare the time series with the task design. Similarly, with the generated model, we can declare the actuation of Tract-DFC and associate them to the white matter locations. Here it comes the challenge. So for each individual can do the fiber tracking, we can use the same parameters. We can set the same number of the tracks, but in the end, they do not necessarily have a correspondence. So this causes a challenge for group-wise statistics. But good news is that now we have the tractogram template, so it's well aligned with the cortical and subcortical gray matter regions, and also the tissue-unbiased brain template provides us a better individual template alignment. Then we propose to inverse swap the tractogram template to the individual space. In the individual space, we can, for each fMRI scan, we can calculate the Tract-DFC for each subject. And so in this way, so the Tract-DFC will have the correspondence across subjects provided by the tractogram template. Then we can do group-wise statistical analysis for the Tract-DFC activation. Again, we tested this idea based on the HCP7 task fMRI. And here we have some results. Here we use the motor task as an example. So you can see that there are different subtasks in the motor task, and we detected the corresponding activation. So this activation pattern agree quite well with the homunculus. And you may ask what is the difference between this activation with the traditional FMI activation. So we did do some comparison. So in the white circle, you're looking at the traditional volume-based activation from the FMI. And we can see that the white matter, the streamline activation in our method not only connect to the traditionally activated regions, but also extends to other brain regions. So this suggests that the functional activation in the brain could be a network work level behavior rather than regional. Yeah, so our Tract-DFC method do provide a new insight of the functional brain mechanism. So in SMM 2024, we did provide more result for different tasks, and we did see quite a distinct activation patterns for different tasks. And so on the right, my student Yifu is working on the resting state Tract-DFC. She's using the ICA method to decompose the group-wise Tract-DFC into different components. And we did see a lot of interesting patterns, interesting combination of fiber bundles. Well, this did show us that there are some functional specificity in the white matter fiber bundles. So, well, combining both the task Tract-DFC and the resting state Tract-DFC, and combining all these results and all these different methods, we're trying to build up a functional atlas of the human brain white matter. So with this atlas, we can use it to infer the functional change caused by white matter damage or white matter impairment. And also it can also be used to inform the neurosurgery or the implantation of the deep brain stimulation or TMS and other interventions. Now let's look at the Sydney subcortical gray matter postulation, another example, another application of our template. This work is led by my colleague, Tonima Ali, and this work is now submitted to Nature Communication. So here, we combine two technology to better characterize each work cell in the subcortical brain regions. So the first technology is the track-weighted mapping. So it uses the tractogram as a media to map different metrics in the brain and refine the mapping. And also with the multimodal template and multimodal registration, we can have the better alignment, better registration for different subjects. And well, in the multimodal brain template space, so we have combined 10 different metrics to characterize the subcortical work cells. And through a clustering approach, we divided the original subcortical regions into 56 small regions. For example, the caudate is divided into 4 regions, and the thalamus is divided into 10 regions. And importantly, we compared our postulation with the histology based postulation. They have shown quite a lot of agreement. This means that this data-driven method with non-invasive MRI really reflect the histology information in the brain, which means that we might be marrying quite the right information about neuroscience in the brain. And with this more detailed subcortical postulation, we can enhance the connectome analysis. So in the paper, we showed an example for connectome analysis of the Parkinson's disease. And with this expanded atlas, comparing with the traditional DK atlas, we did find more important and meaningful connectivities, which is related to the Parkinson's disease. Okay, let's look at the next example. So this work is led by my colleague Marshall Darton. So Marshall is using the multimodal template to look at the cortical hippocampus connectivity. So based on the nice alignment provided by the multimodal registration, they found that different cortical regions preferentially connected to a different subfield of the hippocampus. And importantly, because of the nice registration, we can generate an average map of the cortical hippocampus connectivity map. You can see that for each cortical region, we can kind of generate a fingerprint of the fiber connectivity in the hippocampus. So this is we believe this is quite detailed mapping so far for hippocampus. And now we are also trying to use the track dynamic functional connectivity to segment the hippocampus, because this is a combination of the functional MRI and the diffusion MRI. So we expect structural and functional coherent segmentation of the hippocampus. Okay, now I'm jumping to another example. So we can use the multimodal template to enhance the fixal-based analysis. Well, firstly, I will need to introduce what is a fixal. So fixal is the fiber population in each work cell. So in one work cell, there might be multiple fixals representing different fiber populations. Usually they are in different directions. And with the fixal-based analysis, we can measure the fiber density change, or the cross-section change, or the combination of cross-section and the fiber density change for each fixal. So in this example, my collaborator Sikang Tu used the fixal-based analysis to look at the motor neuron disease. So on the left, with a single model template, we found very interesting patterns, very interesting white matter changes related to the motor neuron disease, particularly the corpus callosum 4 and 5 and the cortical spinal cord left and right are related to the motor neuron disease. But this discovery is quite limited to the core fiber bundles. Now we redid the analysis again with the multimodal template. We found that the finding extends to the superficial white matter, and even in certain regions it extends to the gray matter. So this with the I think this is because of the advantage with the multimodal brain template because it's a tissue unbiased nature, because of it provide because of the template provides quite nice specificity in the superficial white matter and in the gray matter. So it really helps us to provide a complete pathway of the white matter change for the motor neuron disease. And last but not the least, Simon Zanoni, who is a PhD student in our team, is developing the streamline-based analysis based on the tractogram template. So what is the streamline-based analysis? So on the left, we have the work cell-based analysis. Basically, we compare different features in each work cell across subjects or across groups. And in the middle, I just introduced the fixed-based analysis. So we compare the group difference, individual difference in each fixed cell. And now for the streamline-based analysis, the analyzing unit will be the streamline in the tractogram template. So we aim to find the across subject, across group difference on each streamline. So to develop the streamline-based analysis, we are working on a few important technology steps. So firstly, we're working on a sampling strategy, which use the streamline to sample the white matter information or gray matter information. Or actually, it can be used to sample any imaging modalities based on your interest, based on your hypothesis. And secondly, we're developing the similarity measurement between streamlines or the distance measurement between streamlines. And following with this, the similarity measurement, we can do the streamline-wise smoothing, and also we can enhance the statistical inference so based on the similarity or distance measurement. Well, you can see that there could be millions of streamlines in the brain. So if we compare across subjects, there's a multiple comparison issues. So we're also developing some correction method for this multiple comparison issue. So we have submitted our preliminary method framework to ICMM 2025. So welcome to pay attention to our work if you're going to the conference. There are many other applications which is going on or already published. But overall, so the multimodal brain template can be used to map different brain structures, map different brain circuitries. It can be used to study dementia, multiple sclerosis. And worth noting that I have discussed with Kurt Schilling. So there's possibility that our multimodal template can be used to quantify the superficial U fibers, which I believe is very important for brain aging or neuroplasticity. So in summary, so we have proposed a tissue-unbiased multimodal brain template. The template provided quite a unique tissue-unbiased space for neuroimaging analysis, and it is a new framework for method development. And it also enables multimodal fusion. It enables joint gray matter, white matter analysis. And also it's great for joint structure and functional analysis. All the tools I mentioned, for example, the streamline-based analysis will be eventually published as part of the MAtrix toolbox So welcome to use them in the future. So in the end, I would like to acknowledge all the foundings have supported this project. I would like to acknowledge the Matrix 3 toolbox and the community. I would like to thank all the great collaborators and the excellent students who have contributed to this project. And thanks for your attention. I would like to hear your opinions and the questions are welcome.
Thank you very much for your presentation. You started so fast at the beginning, I didn't even introduce you. So
I think I did a self-introduction. All right.
You just went directly. So, well, first, I want to thank you for this great presentation and doing a great job for I don't know how to describe it, like the Pacific time zone of the Tract-Anat Club. So this is very good. And I would like to say also, I love registration-based work. I do a lot myself, like doing my PhD. It was almost a running gag in our lab that I was seeing every problem as a potential registration problem. I really like that. I typically work with bundles. I do population average, but at the anatomical bundle level. And one of the first thing that people ask me is, well, I have very old people or very young people, or I have like groups that are not typically like the average. So they ask like, should we do a new template specific to children? Should we do a new template specific to like aging population? In your case, I think you did your template on HCP data, or like how do you deal with stratifying your data in order to do a somewhat population-specific template?
Well, that's a really nice question, and actually, this is also a question actually we are working on for a solution. Well, I think currently the template we published is based on the HCP young adults data set. But we do want to look at the brain age, the age-related change in the brain. So now we turn to the HCP aging data set. We did find the anatomical difference along the age. So I think one approach is to find a middle space. Like we find the samples with different age and build up a middle space template. And another direction we are working on is to using the generative AI to build up a template that can be changing with age. Yeah, so this will provide the kind of age specific brain template, but there's correspondence across well, different templates. Yeah.
Thank you for the answer. So I went ahead with the first question without even asking if there was a question from the public. So is there anyone in the crowd that has question, comments, something about the application, the technical side? If you want also to write question in the chat, if you're not comfortable talking, I can read them. Is there anyone?
Maybe while we wait, but people get less shy and ask question. Another question that you often get in these type of studies is, that balance between how many subjects you should include to create this template, the balance between putting more and more subjects, potentially blur more the data by more and more subjects, and computational loading that you then is going to take forever. So can you give an idea of number of subjects that you've used for these templates and for the clinical studies, and yeah, so what number of typical size you would be using?
Yeah, that's another good question. I think it will be quite a trade-off. As you mentioned that if we add more subjects to the template, we look more blurry. But we also want to be inclusive. We want to include as more diversity as possible. So far, the current template we released is based on 50 subjects from the HCP HCP project, sorry. but now we are working towards a higher resolution template. We in the new experiments we use the HCP 100 genetically unrelated subjects. So first, I think selecting the subjects will be important. It's better to include more diversity. So the genetic unrelated criteria could help us get the diversity. And also 100 subjects, I think it's a fair number, not too many, not too small. Yeah, I think yeah, so far I believe the HCP unrated 100 is the best number. But for certain studies, for certain small studies, you do not have many subjects. I think to build up a study specific template, perhaps maybe you should at least include maybe 20 subjects. Yeah, and also for certain comparison, it's better so you involve some healthy subjects and also some subjects with the disease. So this will help find the difference across the groups. Also, it's kind of the middle space between different brain and anatomical changes. Yeah.
Well, I'm glad to hear that around 20 subjects seems to be like a good suggestion for like good scale. When I do template with bundles, often I think of 20 and then just to be safe, I double it. Like it's always like a doubling game. Like, I think it's fine with 20 and just in case I take a few more. But averaging bundle, if I have good input and I end up just doing like, what's the average arcuate fasciculus in my population? It seems like same number, 20, 30 people end up like stagnating the shape, and doing more just makes the whole processing a lot harder. So it seems to converge pretty fast with a somewhat uniform population, obviously. And related to that, I have a question. One of the basic like assumption of registration template is that there is like a Gaussian distribution and we're doing the average. Like if I have 60 people that are 5 foot tall and 60 people that are 6 foot tall, and then I do the average, it's 5 foot and a half, but no one is actually 5 foot and a half. Like it assumed that we have a Gaussian distribution. Like, is this true for cortex, white matter? Like, is the assumption of an average person make sense for the brain at the scale of FODF? Do you have a it's a bit philosophical. It's an annoying question.
Well, I think yeah, it's quite a nice angle about the Gaussian distribution. Well, I think yeah, that's a good question. By averaging across the subjects, yeah, there must be an assumption about the distribution. Yeah, I think in most of the time they'll just assume that it's a Gaussian distribution. Yeah, however, I didn't look into the data. I'm not sure whether it is a Gaussian distribution, or yeah, whether 100 subjects is enough to fit this distribution, to mirror this distribution. Yeah. Sorry, I think
I have no answer either. I always
about
assume it. I always think like, oh yeah, of course. Why wouldn't it be? And makes my life simpler. But I never looked at the like. Is it like mathematically sound to do it? I never checked it.
Yeah, but I think to build up a template, we are not doing any quantitative measurement. What we are doing actually is creating the contrast for better alignment. So I think the actual distribution inside each voxel may not be that important. Actually, the boundary itself may be more important for the registration. Do you agree? Like we do not want to quantitatively measure the distribution of each voxel, but we expect that the distribution is different between the gray matter and the white matter.
There is a question in the chat that someone wrote. I'm going to read it. So someone is thanking you for your presentation and is asking if you considered the complex fiber pattern, like crossing fiber, to build your template, and how it would affect the average bundle downstream. I'm also thinking, like, what if one population has, you know, a crossing like this, and another portion of the population has a fiber like this, but then the average is showing a crossing. It's not the same as like having the two separate and then the average shades crossing. How does this behave when you're building your population?
Yeah, I think well, I think this is quite related about building up the tractogram template. When we reach the fiber tracking of this cross fiber regions. Well, I think so far we didn't pay a lot of attention to the crossing fiber locations, but we do want the overall tractogram template to reflect the commonality across the subjects. Well, in other words, our aim is to make the tractogram template reflect the overall or average the connectome. Yeah, so we didn't pay attention to the small regions, but I think based on the FOD measurement, yeah, we did see quite small errors. Because I think this crossing fiber regions are in the major fiber bundle regions. So we did see quite small errors. We see higher or very high angular correlation in these regions. Yeah, I believe that the crossing fibers are well reserved. And also when we register the FOD, we also did reorientation. Yeah, maybe Fernando has more details about the reorientation. Yeah, but I think the reorientation makes sure that the directions in these crossing fiber regions are reserved and are aligned. What do you think, Fernando?
Yeah, no, but I think what you said about the angular correlation is the right thing. So when you compare the angular correlation of the individual data versus the template, it still is very good, even in those crossing fiber regions. So the templates seem to be able to align well those. The contrast that uses the multimodal contrast for the alignment seems to be sufficient to align well those structures because the angular correlation is high there.
Thank you.
I see Kurt as his hand raise. Don't know if Kurt want to go ahead.
Hello, Kurt.
Hey, great talk. I actually have, well, first, also beautiful template. As you know, I've used it, looked at it, and it is absolutely beautiful and easy to use. So
glad you like it.
I had two questions, and they're kind of going back to what Francois was talking about and related to registration. One's on practically using the template, and the second is on variability. So first, so when I use like the traditional MNI 152 template, I'd maybe acquire a T1, I'd acquire some diffusion contrast, I'd use some registration from diffusion to T1, then T1 to MNI. How does that change when using your template? Like, would I change the process of diffusion to T1, or how do I keep the FODs to actually register to, like, basically what contrast would I use to best align with your template? And what do you recommend in terms of software if you want to give a recommendation?
Oh, yeah, I think comparing the two processes, I mean, the single modal registration and the multimodal registration, just to provide more contrast when you do the registration. For example, in our paper, we found that the T1 plus T2 plus MD plus MA gave us so far the best alignment. Then you will need to make sure in each individual, the T1, T2, MD, FA are well aligned in the individual space. Then when you register it to the multimodal template, you provide all these modalities, all the four modalities, and to optimize the registration. Then you will have a better alignment for both gray matter and white matter. Yeah.
So you wouldn't actually use the FOD to drive any of the registration? It sounds like using scalar contrast for the most part.
I think it will be our next step, but if we use FOD, we'll not use the 6 orders of FOD. I think it will. I would recommend to use the low orders at the FOD to for the registration. Yeah. Yeah,
Cool.
but so far I think in our pipeline, I think the T1 plus T2 plus MDFA already gave us a good result. And what you can do is that once you generate the registration, you apply it to the FOD. Yeah, it
Okay.
gives you good result. Yeah.
Okay, that's good to know. And then my second question, and you kind of touched this on the slide, and I guess it's more interesting to the neuroanatomy people. And I'm thinking, when you do the registration, you are currently doing it in the young adult population, but have you looked at where there's the most neurovariability? So I think you could extract that just from your warp fields. And I think that's an interesting question for people studying anatomy, evolution, comparative anatomy, things like that.
Let me share the screen again. Do you mean in this slides?
Yeah, you kind of touched on it with just like the correlation coefficient, but also just the overall some measure of the determinant of the Jacobian or overall displacement of warping would kind of give you insight into like spatial deformation to align to an average space. And I imagine that's different in different parts of the brain.
Yeah, I think this is a good suggestion. Well, I think previously when we built up this template, we focused too much on the commonality. I think we may ignore the variability. That's a good question. I think we can come back to look at the variability, and this could be related to like identifying each subject. So which region is the more kind of reflects the identity of this person, right?
Yeah.
Yeah, it's quite interesting. Thanks for the suggestion.
Yeah, and thank you again.
Yeah.
Jinglei, can I have a clarifying thing based on the first question from Kurt? So you said that the T1, T2, MD, and FA is the most optimal, that the one that you found for the one tested. But for some of the studies that you show, like motor neuron disease or the, I think, the dementia, you didn't have T1, T2, MD, and FA. You just had, what was it, T1, MD, and FA. So can you clarify when, because not everybody would acquire high resolution T1, T2, MD, and FA. T1 and MD and FA, it's more common, but not T2 at high resolution. So can you clarify what you do in those cases?
Yeah, good question. Well, I think to include the T1 weighted and the T2 weighted anatomical scans is because I think T1 weighted image provides quite nice gray matter white matter boundary, and T2 weighted image provides quite a good gray matter CSF boundary, if I'm correct. Yeah, they are kind of complementary information. And also of course the diffusion MRI provides very nice details in the white matter, and all these 3 components contribute to the registration. I think for some studies we only have a T1 weighted or T2 weighted. I think it's okay to just have one anatomical modality, but it's important to have the diffusion modality. Yeah, and of course, most of these studies are for white matter related studies. It must have the diffusion. So if you do not have the complete set of the imaging modalities, just make sure you combine diffusion with one other anatomical scans. Yeah.
Any other question from the crowd? I would have a question still related to bundle. I tried your template before to do whole brain tractogram from it and do bundle segmentation. And it
Mhm.
seems like tractography, especially probabilistic algorithm, expect some noise to exploit it, to explore. And your tractogram, your FOD template is so almost noiseless. It's so nice, it's so smooth that it seemed to reduce the spatial extent of bundle being reconstructed, because probabilistic tracking, it's even harder to look like in the cone of probability, because the FODF are sharp, they are smooth, the crossing are clear, and then the bundle I was getting were smaller in volume than my average bundle, or like the average of my population if I did tracking first. That's not really a challenge or anything. I was just trying it, and I was really surprised by how difficult it was to do a very large exploratory, like, for example, arcuate fasciculus, or doing like massive reconstruction. It was too smooth for probabilistic tracking. I don't know if you played a bit with that, or if you even like know exactly what I meant by exploratory, like bundles.
Sorry, I don't quite follow actually. You mean to generate a large number of streamlines with the template or for a certain structure?
Yeah, I tried like tracking on your template, and I was trying to
Yeah.
get like a nice arcuate fasciculus that cover like a lot of the frontal lobe and a lot of the temporal lobe.
Yeah.
But since your tracking was your FOD were so smooth, the tracking was like the core of the bundle only. It was like the smallest like core with not extra fanning. It was not covering as much like spatial coverage as I was expecting. So I don't know if you observe that or play a bit with that.
Yeah. Well, you mean average FOD will lose some specificity at the superficial white matter or the close to gray matter region, right? Is that your question?
Yeah, it seems like it's harder to exploit the variability of, for example, probabilistic tracking with FODF that are so specific, clearly oriented, so sharp. So that was an observation. I was wondering, like, if you play a bit with that, or if you observe that like some bundle looks smaller on your template than on the subject itself.
Mm-hmm. Well,
Also,
Fernando, is this
something
related to the backtracking technology we are talking about?
in that to some extent, but no, Francois, coming back to you, I don't think we explored that in detail, because all the things that we did were more like, as Jinglei said, on the the track seg of the individual FODs versus the track seg of the template, but that doesn't track. It's track seg doing the track. So yeah, I don't think we have explored that in detail. So it's something would be interesting to look in more detail, yeah. And see
But
if
it
it is.
shows the average bundle in the sense that's like it's the core, like it's the part that everybody has. It's
Yeah,
like
yeah.
really like but yes, I think it's like maybe a specificity issue. Like the it's less noisy, and probabilistic tracking exploit the noise, so it makes sense that it's harder.
Yeah, no, something that we should look into detail, see, and understand a bit more that one. No, we haven't looked into that. No.
It's great. Okay, so now it's 6. So I was wondering if there was more question from the crowd. I don't see any other question in the chat. I see Kurt so said he had one more question. I mean, Kurt, it's 9 a.m. for them. It's fine.
Okay, because I feel
yeah.
asking more if it makes it run over.
No.
Again, I'm going back to Francois's first question, related to templates kind of across lifespan and generalizability to different time points. And I know you haven't, or at least aren't quite there yet, but imagine you're making this for like an aging population or development population. And you mentioned you're using HCP aging or development or one of them, but how do you choose what like scale, what age groups to bin it in? Do you need one from age 12 to 14, 14 to 16, or 12 to 16, or is 0 to 21 or wherever HCP young adult starts good. How would you decide, or can you think of like a smart way to do that to create templates that are useful and meaningful in these age bins? Or is there some metric you'd use to say this is the right number between 0 and 21?
Honestly, now we are working on the HCP aging data set. The age range is 35 to 100. Yeah, so I think one strategy now we are using is to equally sample across the age and use, for example, 60 subjects to build up a template. Yeah, so this will make sure that it's fair middle space. Yeah. But eventually, what we want to do is to develop a age-specific brain template, multimodal brain template. Yeah, which could be, it will depend on some AI technology. We will make sure that each template at each age will reflect the nature of this age, and also it preserves the correspondence across different ages. Yeah.
Yeah, that's what I was thinking of, because you
Yeah.
can imagine like the simple solution is just every decade of life or something. But
Yeah.
creating a continuous template, I don't know what you call it, the 40
Yeah, yes, yeah, exactly. A continuous template. Yeah.
would be really cool and a fun challenge, I guess.
It is. Yeah, but we're working towards this direction. Yeah.
Cool.
Thank you.
Well, I think this concludes. I don't see question in the chat. No one has raised hand. So thank you very much, Jinglei, for your presentation, for the discussion. Thank you everybody for attending. Like I wrote in the chat, next time I will triple check our daylight savings time work in Australia better. So third time is going to be right one. So in March we will have a talk from Maxime Decoteau. There is nothing in February because of the diffusion study group. So I imagine we will have one last talk on the Asia, Oceania time zone before the summer. So otherwise, this was great. Thank you for attending. So see you next time. Thank you again, Jinglei and Fernando, for
Thank
the
you,
discussion.
Francis.
Thanks.
Thank you, everyone. Bye-bye.